the same process that happens. But oh yeah, and so this was really our basis for a lot of research in this field. In 2000, the very first structure was published. This is like a really big deal too. This has like 3,000 or 4,000 citations now. It's the bovine rhodopsin. And if you can cross your eyes, you can actually see it in 3D. I guess I could have given you a 3D structure in video too, but forgot it was a technology is so advanced. Okay, so yeah, this receptor though, they got the structure with something bound to it. This retinol I explained, this retinol here is what binds to rhodopsin. But there's like two types. There's the type where the photon of light changes the retinol from cis to trans. So it's kind of biology speak for just a different conformation of the retinol molecule. And one of them activates this GPCR. It starts that whole signaling cascade, but one of them kind of doesn't do a very good job. Anyway, they got the structure in the one that doesn't do a very good job. So they kind of did, well, they did the best they could do. They got the structure using some complicated techniques, which, oh yeah, I'm actually going to explain. Yeah, to get the structure of anything, particularly you want something that's bound to it, so you understand how those helices are all moving, because they're all moving all over the place. You have to use like crystallography. You have to shine some fancy X-rays at your protein, and those X-rays, depending on what electrons they hit or how complicated your molecule is, they're going to diffract off in different directions. And then you can combine them all together to get an idea of what the protein actually looks like. But it's still a really complicated procedure because you have to make a crystal structure of your protein. And often, like, proteins are so weird and blobby, and they don't stick together in a nice crystal. Like, you picture a crystal maybe as like a nice crystal lattice of like, hexagons or something, but it's really tricky with proteins. So the goal is to crystallize all these membrane proteins, and it's even harder because they're in a membrane, and you have to get rid of the membrane if you want to get a protein structure. So how do you get the beta-2 adrenergic receptor, which is it's the new one to tackle. It's like this was the idea, was to see what's different between this adrenergic receptor to the one in your eyes, the rhodopsin. So it's tough because, yeah, I explained that they're kind of blobby and tough. The wild-type beta-2 adrenergic receptor, you can't crystallize it very easy. There's not that much of it. The intracellular loops, that's like the stuff in between the helices that's floating around on the inside of the cell, moves around a lot, and there's a lot of flexibility. And yeah, there's a breakdown of all these loops, and your thing falls apart. And there's also there's not very good contacts to make a crystal. To make a crystal, you have to have polar contacts so that, well, I don't know. You have to have contacts at various points around it if you want to make a lattice of them. So this is just a copy. This is 1, 2, 3. They're the same, but they have to have contacts between them. So the big breakthrough, but maybe not, that's probably been done before, was to modify the wild type receptor, change it up a bit. So you slice off some of the protein and you add some in order to try and make some contacts. So you can actually take the amino acid sequence, take a chunk out, and then throw your own in there, as long as you kind of have a good idea of what to put in there. And you can hopefully get something that can become a crystal. So they, in particular, this paper, they use this lysozyme. That's just like stuff you find in egg whites or whatever. But it just happened to fulfill a bunch of criteria. So actually, in particular, like I told you here, that the big problem was this one particular loop of our protein. I should show you the structure and show you exactly where it is, but you'll see it later. Anyway, that was the big problem. So they sliced away that intracellular loop, and they replaced it with some lysozyme. And so it added a nice polar surface, and it made sure that our helices didn't move around too much. And well, I think that worked out pretty good. So, oh yeah, this is a diagram of just the area where they sliced off and then they inserted it there. They also sliced off some of the end of the amino acid sequence. Here's just like a diagram of, yeah, just where they sliced it. This is the intracellular loop 3. And this is helix. This is just one of those helices until the 7. But in particular, this one was the problem. Okay. I don't know what my time is, but I actually gave this as a 40-minute talk, but I tried to condense it. Anyway, I'll just go on. Yeah, so what's next? I forgot. Anyway, oh yeah, we figured it out. It worked. So if you cross your eyes with this one, you can see that that's the part we inserted. And these are sort of like lipids, because it's in the cell membrane. They kind of just stuck around. And we noticed some interesting things from the crystal structure is that something that the rhodopsin one never had was this extracellular loop up here. And this is the area up here where all those ligands bind, all those interesting, all the dopamine and stuff, all goes up here. And that's got to affect how the binding works to this protein receptor. Okay. So the idea is that, or the question you have to ask, it's just natural question, is we modified our structure so much, it must be different. It must be completely different. Well, they did this sort of test, whether to see this made-up beta-adrenergic receptor with lysozyme did the same job. And it had similar binding affinities, so that it just reassures you that the changes we made to this receptor didn't end up being a big deal, except for the fact that it didn't actually bind to G proteins, because that big lysozyme was stuck in there. But we're not really interested in that part for now. We're kind of interested in where the ligand binds, and I guess that's still okay. So this is, I guess this is kind of drug talk, which you should probably know, is that there's like a baseline activity for these receptors. So you don't do anything, you just have a normal physiology, normal system. It just does some baseline biological response. So that pathway gets turned on at some basic rate based on whatever the normal things that bind to it. Anyway, there's other things that bind to it that make it stop, or make it do a really bad job. It goes to 0% biological response, something called inverse agonist. A neutral agonist is something that fills the spot. It binds, it's a ligand that binds to this receptor, but it doesn't do anything different. It just prevents other things. It prevents these inverse and full agonists from binding to it. It just kind of, it's a blocking. It's cock block. But there's also things that make it work really great. A full agonist or partial agonist, which really improve the effectiveness of this receptor. And so this is really the range of things that can bind to your receptor, and you want to design drugs to do these things. So yeah, things bound to it. These are things they tried to bind to it, salbutamol, and I think these curves are just represent before and after the change. Oh yeah, so the blue line is after they made the change. Okay, the function, yeah, ligand binding. So they crystallized, they got this protein structure with something bound to it. What was bound to it was this thing called carazolol. And itself, it's a partial inverse agonist, so it kind of does a bad job. It's kind of like it's a lot like the rhodopsin structure where I said it's kind of a bad structure too. They're both inactive states. There's an inactive state of rhodopsin, and this is an inverse agonist. It's also an inactive state of the adrenergic receptor. Which is, it's good so you can compare them, right? We want to be able to compare the inactive state of one with the other. But it also really begs the question, obviously, about what does the active state look like? How does all these helices move when something activates it to whatever, full 100% response? So we have that. How can we, yeah, we want to compare it. This is a comparison. On the left is the beta 2 adrenergic receptor.